The study of plants reveals an incredible diversity of forms and functions. To understand this diversity, botanists often turn to reproductive characters. These are the features related to how a plant creates the next generation. They are fundamental to the survival and evolution of plant species. These characters include the structures of flowers, the development of fruits, and the formation of seeds. Unlike vegetative characters such as leaves or stems, which can change greatly due to environmental conditions, reproductive features tend to be more stable. This stability makes them exceptionally reliable for identifying and classifying plants into related groups. Reproductive characters provide a detailed blueprint of a plant's evolutionary history. The intricate parts of a flower, for instance, tell a story of co-evolution with pollinators like insects, birds, or even bats. The way seeds are protected within a fruit reveals strategies for dispersal, whether by wind, water, or animals. Examining these features allows us to see the solutions that different plant lineages have developed to solve the universal challenge of reproduction. From the smallest wildflower to the tallest tree, these characters are the key to understanding the lineage and relationships that connect all plant life on Earth. The consistency of these reproductive traits is why they form the backbone of plant taxonomy. When we compare the flowers of two different plants, we can make educated guesses about how closely they are related. For example, plants in the same family often share a similar floral structure, even if their leaves or overall growth habits look very different. This focus on reproductive biology helps bring order to the vast and sometimes bewildering world of plants. It allows us to build a classification system that reflects true evolutionary relationships, a system that shows how different species have diverged from common ancestors over millions of years. Therefore, when we explore the world of plant reproduction, we are doing more than just looking at pretty flowers or interesting fruits. We are delving into the very mechanisms that drive plant evolution and diversification. These characters are the language plants use to ensure their continuation, a language that botanists have learned to read to unravel the complex tapestry of life. Understanding these features is essential for anyone interested in the biology of plants, from the amateur gardener to the professional researcher. It is the starting point for appreciating the elegance and complexity of the plant kingdom. The flower is the centerpiece of reproduction for a vast number of plants known as angiosperms. It is a specialized shoot system designed to facilitate fertilization and produce seeds. A typical flower is composed of several key parts arranged in concentric rings or whorls. At the base, we often find the sepals, which are leaf-like structures that collectively form the calyx. The primary function of the calyx is to protect the unopened flower bud. In some plants, the sepals can fuse together at their base, forming a structure known as a calyx tube. This tube can provide additional protection and structural support for the developing flower parts within it. Moving inward from the calyx, we encounter the petals, which together make up the corolla. Petals are often brightly colored and scented to attract pollinators. Inside the corolla are the reproductive organs themselves, which are essentially modified leaves called sporophylls. There are two types of sporophylls in a flower. The stamens are the male sporophylls, each consisting of a filament and an anther where pollen is produced. The carpels are the female sporophylls, located at the very center of the flower. A carpel, or a group of fused carpels, forms the pistil, which contains the ovary, style, and stigma. The stamens represent the microsporophylls, as they produce the small spores known as pollen grains. Each pollen grain contains the male gametes. The carpels, on the other hand, are the megasporophylls. They enclose the ovules, which are the structures that contain the female gamete, or egg cell. After successful pollination and fertilization, it is the ovules that will develop into seeds. The arrangement and structure of these sporophylls are incredibly diverse across different plant species and are critical features used for identification and classification, reflecting different evolutionary pathways for successful reproduction. The overall architecture of the flower is a testament to its function. The arrangement of sepals, petals, stamens, and carpels is not random, but is finely tuned to ensure reproductive success. The calyx tube, for example, can influence which pollinators can access the nectar, while the position of the stamens and stigma is often optimized for efficient pollen transfer. Every part of the flower, from its protective outer layers to its central reproductive sporophylls, plays a vital role in the complex process of creating new life, making it a true marvel of biological engineering. Inside the flower's ovary lies the future of the plant, the ovules. The specific arrangement of these ovules within the ovary is known as placentation. This term refers to how the placentas, which are the regions of tissue where the ovules attach, are distributed. The type of placentation is a very important and stable characteristic used in plant classification because it is genetically determined and less influenced by the environment. It provides crucial clues about the evolutionary history of a plant group. Understanding placentation helps us understand the fundamental structure of the ovary and how it has evolved across different plant families. There are several distinct types of placentation, each with a unique configuration. One of the most common types is axial placentation. In this arrangement, the ovary is divided into multiple chambers or locules by partitions called septa. The ovules are attached to a central column where the septa meet, much like segments of an orange. This is commonly seen in plants like tomatoes, lilies, and hibiscus. Axial placentation is considered a relatively advanced trait and provides a well-organized structure for protecting and nourishing a large number of developing ovules within a partitioned ovary. Another significant type is parietal placentation. In this case, the ovary is not divided into chambers. It is a single locule. The ovules are attached to the inner wall of the ovary. This arrangement is typical in plants like orchids, violets, and cucumbers. It allows for the development of many ovules along the extensive surface area of the ovary wall. A third type, free central placentation, also features a one-chambered ovary. However, the ovules are attached to a central column that arises from the base of the ovary and is not connected to the ovary wall by any septa. This is found in primroses and carnations. Other types include basal placentation, where one or more ovules are attached at the very bottom of a single chambered ovary, as seen in sunflowers and buttercups. Conversely, apical placentation involves ovules attached at the top of the ovary. Each type of placentation represents a different structural solution for housing and supporting the ovules as they mature into seeds. The study of these arrangements is not merely an academic exercise. It is a fundamental tool that allows botanists to accurately identify plants and reconstruct their evolutionary relationships, revealing the deep history written within the flower. In many plant species, flowers do not grow singly, but are grouped together in clusters. This arrangement of multiple flowers on a single stalk is known as an inflorescence. The evolution of inflorescences is a significant strategy for enhancing reproductive success. By grouping flowers together, plants can create a more conspicuous visual display, making them more attractive to pollinators from a distance. This clustering can also maximize the efficiency of pollination, as a single visit from a pollinator can result in the fertilization of many flowers at once. The structure and type of inflorescence are highly varied and are key diagnostic features in plant taxonomy. Inflorescences are broadly classified into two main categories based on their growth pattern. The first is racemose, where the main axis continues to grow and produce new flowers, with the oldest flowers at the base and the youngest at the tip. This pattern is called indeterminate growth. A classic example is the raceme, a simple inflorescence with a central axis bearing stalked flowers, as seen in lupines and snapdragons. A spike is similar to a raceme but has flowers that are sessile, meaning they lack stalks, as observed in plantain. The familiar cattail and the spadix of an arum lily are other specialized examples. The second main category is chymosy inflorescence. In this type, the growth of the main axis is terminated by a flower, so it is said to have determinate growth. Subsequent flowers develop on lateral branches below the terminal one. This results in the oldest flower being at the center or top of the cluster, while the younger ones develop outwards or downwards. A simple sign can be seen in plants like chickweed or phlox. The arrangement ensures that the central first opened flower is most prominent, which can be advantageous in guiding pollinators. This growth pattern creates a very different overall shape compared to racemose types. Beyond these basic types, there are many complex and specialized inflorescences. For example, the head or capitulum found in the sunflower family, Asteraceae, is a highly condensed inflorescence where numerous small flowers called florets are packed onto a common receptacle. What appears to be a single large flower is actually a composite of many. Similarly, an umbel, characteristic of the carrot family, Apiaceae, has numerous flower stalks of equal length that arise from a common point, like the ribs of an umbrella. These diverse floral arrangements are not just beautiful, but are functional adaptations. And studying them is crucial for understanding plant relationships and their evolutionary journey.